Okay, hi everyone. Today we have another session of the Sybeco, and today we are hosting Balint Kiraly from Budapest. Balint originally studied physics at the Budapest University of Technology and Economics. He completed his master's in medical physics, and during that he worked on in vivo imaging techniques. During that time, he became fascinated with neuroscience, which led him to pursue a PhD in neuroscience in the lab of Balázs, Hangya. His research focused on the interaction of neuromodulatory systems in shaping different learning processes and in underlying neuronal dynamics. He finished his PhD with Balázs and then moved to Vienna. He received the EMBO fellowship, and he's now starting his postdoc again with Balázs. And I'm not sure what your postdoc research is supposed to be on. But he's for combining approaches from machine learning, data science, and physics to study how these processes work across health and disease. Thank you, Balint, for accepting our invitation. We're excited to have you here.
Also, thank you for the opportunity. I'm very happy to talk about neuromodulatory systems, which is also going to be my postdoc topic on the future too. And now I'll focus on the cholinergic and the dopaminergic systems. Yeah, so and that's because in a very nice analogy with how engineered systems can learn from trial and error, one of our most consolidated theories of learning in the brain is the dopaminergic influence on learning from reinforcement. So from the work of Wolfram Schultz, Peter Diane, and Reed Montague in the 90s, we know that dopamine scales with the so-called reward prediction error, which is the difference between experienced and expected reward. When a monkey received an unexpected reward, they noticed that midbrain dopaminergic neurons increased their firing. When this reward got fully associated with a conditioned stimulus, this activation propagated to the time of the predictive signal, and later when a predicted reward was omitted, the dopaminergic firing was suppressed. Reinforcement learning algorithms can use exactly this signal to update the value function and the agents model of the world. This fundamental finding influenced neuroscientists in the past decades to interpret other neuromodulatory systems in orthogonal terms. For example, a prominent example is the proposal of Kenji Doya that we could interpret other neuromodulators along distinct reinforcement learning variables. For example, the learning rate for acetylcholine or the exploration trade-off with noradrenaline. However, recent findings, including our own work, revealed that other neuromodulators also tend to signal outcome expectations. We have shown that short latency cholinergic firing after feedbacks in a probabilistic Pavlovian conditioning can be relatively accurately explained by an unsigned prediction error model, likely resembling salience. So similar to dopamine, surprising rewards evoke a bigger response from cholinergic neurons, but this unsigned prediction error is practically the absolute value of prediction error. So negative surprising feedbacks, such as punishments, are also evoking a positive response. This and other findings, for example, the work of Jeremiah Cohen or Zach Maynan in other neuromodulatory systems, raises a fundamental question that do these neuromodulatory systems encode indeed largely independent variables as thought for a long time, or do they instead convey overlapping information, perhaps differentiated primarily by their distinct downstream impacts. It's worth to note here that the majority of the cortex receives much denser innervation from other neuromodulatory systems than from the dopaminergic system. To address this, we had to overcome some technical challenges. So first, one possibility is that these similarities arise from overly controlled Pavlovian task designs, which were common for a long time. Therefore, we designed an operant framework that dissociates rule learning from contingency learning. We do this by training mice to learn an auditory go-no-go association task, where animals can obtain voter rewards by answering to cues and avoid airpuff punishments by not answering to other cues. One go and one no-go association is kept fixed throughout the entire training process, so we massively overtrain animals on these control contingencies. But on the backdrop of stable performance in these associations, we introduce novel cue outcome associations, either go or no go. They are changed in blocks every time the animal learns this association. This task led us to these nice psychrometric learning curves, because in this task we can control the task difficulty by the similarity of these new cues in their frequency to these fixed overtrained pairings. So, for example, a novel go tone, which is very similar to the fixed go association, is very easy to learn. Animals can learn it in a few trials. On the other hand, when it's a no-go tone, which is very similar to the fixed go tone, it's a massive challenge for the animal to form this association.
Question, Balint.
Yeah.
So you changed these tones across blocks of trials, or how
yes, yes. So these two tones are kept fixed through the whole training. It can take 2 months, through whole time fixed. But in blocks of maximum few hundred trials, there's always 1/3 new association, and both the frequency and the outcome, the predicted outcome of this new association is randomly switched between blocks. And we switch blocks when the animal learns the association, by which I mean that the animal reaches 70% performance in all of the contingencies.
So usually in one session, how many different tones you manage to test?
It's from 1 to 6, depending on whether they are randomly easy or not, and also of course on the animal.
Right, and then so for 2 months they learn they have that basically pre-testing with the same tone. And then how many of these testing sessions you have where you introduce novel tones?
It's so first we by step by step learn the rules of the task, that there are go tones and no go tones. That's lie around a week. Also varies across animals, and then for several weeks we train the animals on these new associations up to 6 weeks.
What I'm trying to understand is that if there is no repetition of that initial tone, then when you compute distance from the initial tone, that initial also keeps being updated, because now the mouse may start using the tones, these novel tones, the similarity to those, unless you have a baseline block in every session that reminds them, oh, these were the initial tones that you got trained on for 2 months.
No, so, yeah, sorry, I missed that. So that all the time, around 1/4 of the trials is these fixed go tones. So actually it's balanced. We always have 50% go and 50% no go tones,
right.
And depending on whether the novel cue is a go or a no go tone, it is 50% balanced between the old fixed and the new cube.
Right.
So practically all the time, 25% of the trials are de novo associations.
And you start with the old one, and then you introduce the novel.
No, they are mixed. So they are randomly mixed.
Okay.
If
So
that's what you mean.
Yeah, it's a little bit confusing. It would be very helpful to have a diagram that shows in a session how these different old and novel tones are presented and the block structure. But I think I get the general message.
Yeah, I'll prepare one. But yeah, practically the general message is that it's randomly drawn with these probabilities. Every trial is 25% to be a novel association, the current novel association, and 25% to be the same outcome fixed association, and 50% to be the other type of association. Okay. Yes, and our other aim was, or other task was to, if we want to understand the relation of these systems and how they cooperate or compete, then we thought that we have to monitor these systems simultaneously. Relatively recent neuromodulator sensors and fiber photometry made this task significantly easier, but you will later see that to go to the core of this question, you still need to record single unit activities to be able to disentangle all the details of these interactions, which was a massive challenge. I probably would have never succeeded without my very enthusiastic undergrad Vivi, and we also had to develop an in vivo localization pipeline to be able to verify the success of these surgeries in the live animals and not just from the post hoc recordings, from the post hoc track reconstructions.
How did you do?
Yeah, so both before and after surgery, we made micro CT and MRI images of the animals. To be more precise, we only did MRI images before the surgeries. And on the CT images, you can beautifully see the electrodes. You can actually even see it in this 3D micrograph. And we co-registered these images to find out that whether our implant indeed managed to hit our tiny deep brain targets, which were the basal forebrain and the midbrain HDB and VTA. Yeah.
Do you have any idea why not that many neuroscience labs use these CT and MRI for mice for localization? It's not common. Most people do post-hoc histology.
I think one thing is that this really worth it when you have a very demanding experiment. Like for us, it worth it because we were then training these animals for months and not doing a one-day experiment. The other thing, I guess, is that it's probably rare to have both CT and MRI available. Especially CT is not very common in neuroscience institutes, I think. But otherwise, I can definitely suggest it. So I think it's worth it, especially if it's available. I don't know whether it worth to buy a CT just because of this, and you need a good resolution CT, at least for small implants, and these implants are usually thin. Yeah.
And these are specialized for rodents, or?
Yes, exactly. Yeah. So it has to be a micro, so-called micro CT. Yeah. But we were lucky because my previous lab and my master's work was in the lab where I was working with these equipments. So we recorded simultaneous dopamine and cholinergic and dopaminergic activity. So this is a very busy figure. Let's go step by step. So let's start with dopamine release measured with fiber photometry from the ventral striatum. We're going to see something very similar to the monkey experiment in the introduction. So in this task, there are so I partitioned trials from the task to different categories. Hard rewards are called dose. In this session, there was a new go tone association, and we called those rewards that were followed by this yet still under learning new go tone. So these rewards were not that much expected by the animal, and not the association was not that strongly formed, and these rewards evoked the largest response from the largest dopamine release in the ventral striatum. Easy rewards following the overtrained fixed go tone evoked a much smaller release of dopamine, though on the other hand the cue predicting these easy rewards also evoked a robust release of dopamine. In those trials where the animal made a mistake, so there was a no-go tone, but still the animal answered, was expecting to get a reward, but got a punishment instead, then the dopamine release was below baseline. When we examined the acetylcholine release in the BLA, we found something qualitatively very similar. So hard rewards evoke the largest release of acetylcholine, while very well expected rewards evoked much smaller, close to zero release from acetylcholine, but predicting tones evoked a massive release of acetylcholine. And the main difference that punishments also evoked a positive, an increased release of acetylcholine. When we examined the single unit activities, we found something quite similar in the level of the single units.
How did you do this? So the left side is fiber photometry, and the right, what did you do here?
The right is tetrode recordings with optogenetical tagging.
I see.
So we crossed CHAT-CRA and DAT-CRA mice, so we were able to tag cholinergic and dopaminergic neurons in the same animal from the basal forebrain and the midbrain. And the same story. Less expected feedbacks evoked large positive cholinergic firing, as well as especially fixed go tones. The difference was that, in line with a series of papers from the past decade, dopaminergic neurons formed a more heterogeneous population. They were homogeneous in coding reward expectation and the unexpectedness or the expectedness of reward. So this component of reward prediction error, and around half of the dopaminergic neurons were very similar to the nucleus accumbens, the ventral striatum dopamine release, and to the example in the introduction, by which I mean that after punishments these neurons were robustly inhibited, but around the other half of the dopaminergic neurons were qualitatively more similar to the cholinergic activity pattern. So they were also activated with a short latency activated after punishments.
The punishment that you referred to from the classic Schultz, so that is the unexpected absence of reward. But in this case, it's the no-go.
It's an actual.
So,
yeah, here
exactly.
it's an
so
actual
I wouldn't have expected. I wouldn't have necessarily expected a decrease of dopamine here.
Why I expect decrease of dopamine here is that this is an operant task. So practically the animal, if the animal answered to the cue, then it's most probably because the animal is expecting a reward. So at the same time, it's also an omission of reward. I think so. Yeah.
But I'm saying if the mouse has learned the association that the no-go tone is not going to be followed by reward, then why would dopamine neurons show a negative reward prediction error?
If the animal learned that a no-go tone is a no-go tone, then why the animal answered to it? So probably
unless
the
you're
animals
saying
are
that
not...
they licked here. So you're saying from their licking behavior, you read out that they had the internal prediction that there's going to be. So these are these are the when you say punishment, are these correct rejections? What are these? These are
punishments
false alarms?
are false alarms. Yes.
Okay. So, then that makes sense. That makes sense then. Yeah.
Sorry. Yeah, but I also read that out from the. So I also checked the leaking behavior too. But yeah. So these are false alarm trials. Yeah.
And if you look at correct rejections, I suppose how do they look like?
I can actually
because that's
show
where
you
we wouldn't expect
that.
the negative response.
So here you can see correct rejection times, and interestingly, correct rejections are followed by a brief activation of all three types. Both cholinergic neurons, type 2 dopaminergic neurons, and type 1 dopaminergic neurons.
But it matches the hypothesis that we don't see that negative signal here anymore because the absence of reward is not.
Yes.
Yes.
Yeah. Even better than that. So how I would interpret this is that when the no-go tone ends, then the animal is probably actually relieved that the animal that it managed to escape the punishment, or managed to withhold licking for long enough to not get the punishment. So there's even a small activation of type 1 dopaminergic neurons. Yeah. So I'm gonna refer to these type of dopaminergic neurons, which are suppressing their firing after punishments as type 1 dopaminergic neurons, and these type of dopaminergic neurons, which are qualitatively similar to the cholinergic activation pattern, I'm going to call them type 2 dopaminergic neurons. Yeah. So this task design allowed us to have, I think we identified around 8 behavioral readouts which are related to reward prediction error. So first, we checked that if you remember from the previous slide, task difficulty was controlled by the similarity of the novel association, the frequency of the novel cue, to the same outcome fixed association. And we observed that both of these systems in their cue responses very nicely represented this feature of these cues, of how easy were they to predict rewards from these tones. And we partitioned trials along other behavioral aspects too. So, for example, we examined learning progress based on the mice new cue responsiveness. So we partitioned trials to new cue trials when the animal was not much responsive to the new cues, when the animal was responding around 50% of the trials to the new cues, and when the animal formed the new association in case of go trials, or the opposite direction in case of no-go trials. And again, both systems very similarly represented this reverse predictive inner value of these cues to the animals.
Question. So how do you measure similarity across tones?
It's measured as the frequency distance, the distance in frequency.
So these are just pure tones, and you see how
yes, these are
how close the frequencies are.
Yes, right? yes. Yeah. And we checked 6 other aspects, including, for example, reward expectation. So it's the same now. Trials are partitioned between based on, again, the responsiveness of the animal to the new cues, and when the animal starts to form a go association, it evoked a somewhat larger cholinergic and dopaminergic response to rewards, when these rewards were not that much expected yet, and five other aspects, including the temporal uncertainty of rewards, choice confidence measured as the reaction time, the punishment avoidance, which I have shown you in the previous question. The long story short is that we didn't manage to find any aspect in this task where we would have found an obvious difference between the prediction error coded by these systems, except for the first observation I made, which was that around half of the dopaminergic neurons were suppressed by punishments, in contrast with the other half of the dopaminergic neurons and cholinergic neurons. But other than this, we found an unexpected, very robust degree of signal correlation between these systems, which further raised the question in us that if these systems code such a similar, that seem to code such a similar information in the brain, then what connection they can have. So we
question, Balint, before you move on.
Yeah.
So I get the general message that they're similar, but there is something I'm not following here. On the left side plots, let's focus on just the top row. So the cholinergic and dopaminergic seem very different. Dopaminergic is reward driven, and cholinergic seems to decay slowly before the reward. So, but I thought you showed us that, but these are also very different from the top row on the right side. That shows cholinergic is reward, is punishment. Yeah, well, that is punishment. So don't worry about that. Let's focus on the top left. There is a very different response profile here.
Yeah. So usually we don't have too much trials here, and that's why these average curves are quite noisy. The reason for this, so actually you find the other difference, which I was saving for later, is that interestingly, while feedback responses are extremely fast in cholinergic neurons and faster than dopaminergic responses, cue responses were relatively slow compared to dopaminergic cue responses. So the opposite sorting. Right. And it's quite often that there is only a small window between the rewards and the response of the animal. Another thing is that the cholinergic response is quite often a single action potential, or maybe two action potentials, while cues evoke a relatively bigger response from cholinergic neurons. So in these kind of aspects, there are differences between the systems, but there are differences across neurons within the systems too.
Right. Yeah. But then why is it that the average reward for cholinergic in the slide that we were just at looks different from here? This looks very different from the 1, 2, or 3 slides before.
Yep.
Yeah. Here or the next. Yeah. Because
I don't think it looks terribly different. So if you just
I see
check
here is that from 200 milliseconds, well, maybe it's a different time scale.
Yeah.
Okay. Yeah. All right. We don't, but I mean, but just roughly looking at these, one is decaying, the other one is responsive. So that's why I'm saying, but we can move on.
If you check, then in those trials where the animal already formed the association and responses in a large percentage of the trials, then this curve is quite similar to this curve here, the light green curve here, which is the fixed association. So if you check here, of course, it's a much wider time scale, but you're gonna see a decay of the light green curve, which is because there is a large activation following the fixed go cue, and this is practically what we see there too. That the already formed association evokes not much reward evoked response from the cholinergic neuron, so that curve is largely dominated by the previous cue response. If you check only the dark green curve, then I admit that this is noisy because I don't have much data in this. But if you check this curve, then actually there is a
I guess the dark and bright green also are not the colors are not very easily. Okay.
Okay. Yeah. So we found an unexpected degree of signal correlation between these systems. So we went further and we tried to understand the relation of these signal correlated systems with each other. First, we tried to predict behavioral new cue responses of the animal from the cue responses of the basophoric cholinergic and the dopaminergic neurons. And again, we found that both systems were quite good, quite capable to be these. We are talking about single unit responses, but still were good predictors of the animal's response, and they were not significantly different. But when we combined the cholinergic and the dopaminergic cue responses, we were able to make significantly better predictions on the animal's response. And this is not just a trivial effect, which I want to show you by also comparing this to predictions based on the combination of the two dopaminergic subtypes, which were not much better than predictions based on single dopaminergic neurons, but the combination of the two systems were had a robustly better predictive power than just dopaminergic combinations. So even though they are very similar and strongly positively correlated, they seem to be not fully redundant. And then so far we also I didn't went into details on this, but we also haven't found much difference between the two dopaminergic subtypes neither. So both subtypes were coding reward prediction error, with the exception that these neurons were suppressed after punishment and these neurons were activated after punishment. To further understand these coding relations, we checked partial correlations in the cue responses of cholinergic and dopaminergic neurons and the behavioral responses of the animal. And not surprisingly, these when we controlled for either dopaminergic, type 2 dopaminergic variance explained by type 2 dopaminergic neurons or by cholinergic neurons or the behavior, these correlations were weaker than the zero order correlations. This is not surprising, as I have shown that these codes are strongly correlated. Surprisingly, we found a difference between the two systems. So in contrast with this symmetric relation between type 2 dopaminergic and cholinergic neurons, the cholinergic neurons had an asymmetric relation with the type 1 dopaminergic neurons in this partial correlations. So while controlling for the cholinergic, the variance explained by cholinergic cue responses decreased the correlation of dopaminergic cue responses and the behavioral responses. This was not the case in the cholinergic neurons correlation with behavioral responses when controlled for type 1 dopaminergic responses.
Question, Balint. I'm not sure. So the left side was clean that you're showing us when you basically combine both the cholinergic and the dopaminergic, you do see better prediction. So it cannot be fully redundant. I'm not following the right side. What is zero order? What you mean by controlled? So can you maybe take one of these plots and tell us what that is about and what the message is here on the right side?
Yeah. So this box plot shows the distribution of the correlation coefficients between the new cue responses of basal forebrain cholinergic neurons and the behavioral response trend of the animal, whether it answers to new cues or not. And I would expect, based on everything I said before this, what you can see in all the other figures. So for example here, that when you compute the partial correlation where you correlate here the cholinergic responses with behavioral responses controlled for the information explained by type 2 dopaminergic neurons, then if there is redundancy in this system, some redundancy, then we would expect that the correlation coefficients will be weaker, as both systems are good predictors of the behavioral responses.
Mm-hmm.
And we see this all the time. We see it also here, that dopaminergic correlations with behavior.
When you say controlled, what do you do in your analysis?
So I compute partial correlation between, let's say here, cholinergic neurons and behavior as a
Yeah, it's okay. Yeah. But then the main point is that once you remove it, once you control for it, if it was redundant, it should not change the R. However, if it changes the R, that means that it was not redundant. Is that the right thing, or were you saying the opposite?
I kind of saying all here. So they are to some degree redundant, but not fully redundant. The point rather here is that there is an asymmetric connection here. But I think I'm going to have better proofs for this soon. Yeah. Yeah. So the idea, this raised the idea here that that there might be a directional influence between the cholinergic and the type 1 dopaminergic neurons. So we fitted a lagged linear regression to use cholinergic cue responses to explain the behavioral response trends of the animal. But we did this by allowing temporal shifts to be added to the neuronal signal. So we also allowed that the future of the cue responses would explain the current or the past of the current behavioral responses. And then we examine that which time lags allowed us to have the best fits between neuronal responses and behavioral responses.
And when you say behavior, it's fitting with what? Is it like the D prime? Is it the licking? What exactly you're correlating?
It's we're correlating it with the trend of responding or not responding to the new cue. Okay. So it's a moving average, a small moving average window on responses.
On the hit responses, so it's a moving average on hit responses. That's so it's all about hits.
Hits or so go or no go responses. Yeah. So.
Right. But like false alarms.
It depends on whether it's currently a go or a no-go tone, but the behavioral readout is binary, yes or no. But we have a moving average on this. Okay. That's what I call a trend.
Okay.
The point with this approach is that we find indicators that the basal forebrain cholinergic neuron responses led both behavior and both the type 1 dopaminergic responses. So, for example, here we show that a few trials earlier cholinergic responses fitted or explained best the current trial dopaminergic responses. So cholinergic cue responses tended to anticipate both the behavioral trends and the type 1 dopaminergic trends, while this was not the case for the other. Yep.
But I thought you showed that the cholinergic responses to the cue were more delayed.
Yes, exactly. So in within trial, you're right. And this is I think this is very interesting. So within trial, that's the order of responses, that first actually type 1 dopaminergic responses, then type 2 dopaminergic responses, and then at the end cholinergic responses. Now what I'm talking about is a level broader. So now I'm talking about that across trial, how these trends work. So the cholinergic neurons are a better indicator of that the animal across trial starts to respond more to a go tone or starts to respond less to a new go tone. Type 2 dopaminergic neurons are in a nice synchrony with the current responsiveness of the animal, of whether responding or not. And type 1 dopaminergic neurons tend to, with a small lag, a single trial lag, but tend to lag the behavioral responsiveness of the animal.
Then are you saying that the response in cholinergic neurons predicts the subsequent trials behavioral response?
Yes. With a very short latency, one trial, but yes.
Right. I see. It's the lack of trials. I just saw your Y label. It's not time. Okay.
Yeah. What is much more robust is this, that that the cholinergic Q responses anticipate what dopaminergic neurons going to how dopaminergic neurons going to respond to new cues in subsequent trials.
Yeah. But this doesn't match the.
No, yeah, the within trial, the inside one trial order.
No, it doesn't match the idea that if dopamine is doing temporal difference and cholinergic is doing learning rate, then you would expect the temporal difference to come first.
Yes.
And then modulate the learning rate.
Yes, probably yes. Yeah.
Or maybe no. I mean, they can be independent.
Yeah. And it's also, I think it's non-trivial how learning rate operates.
Or maybe because learning rate is a function of the certainty, then that reflects more the context. So that's first modulated, and then we have these errors more on a trial, like more reflecting the single, like within trial.
Yes, and it's also a question of when do you need a learning rate boost, because you might want to increase your learning rate based on feedbacks rather than based on cues, and in that sense the cholinergic responses are the fastest. So it actually makes very much sense that when there are unexpected feedbacks, regardless of their sign, whether positive or not, to increase your learning rate, to boost your learning rate, because the model is not good. There are big unexpectednesses. But probably the picture is not that easy. That acetylcholine is learning rate and dopamine is temporal difference reward prediction error. So there are much stronger correlations here than what would be expected based on independent variables. So but we kind of went through the same questions. And therefore, we try to understand what can be the exact relation of these systems. So we went one step further to go from these trial averaged and trial by trial trends to moment by moment correlations. So we went for the most direct measure to be examined between these firing activities, and as we had single unit recordings, we were able to compute cross correlations between cholinergic and the two dopaminergic neurons. And what we found was a complete segregation of interaction patterns between the cholinergic and the dopaminergic systems, a complete segregation along the subtypes. So type 1 dopaminergic neurons, those which are suppressed after punishments, were suppressed after cholinergic neurons in general. During the task, out of the task context, in intertrial intervals, the general trend was that type 1 dopaminergic neurons fired less after cholinergic action potentials. On the other hand, type 2 dopaminergic neurons showed a zero lag positive correlation with the cholinergic activity. Type 2 dopaminergic neurons tended to be slightly more active when
these are simultaneously recorded?
Yes.
Right.
Yeah. Yes. And this is simultaneously recorded fiber photometry recordings, which is interesting also to some degree showing a double nature of correlations, a positive around 0 or slightly before 0, and a negative correlation after 0. But let's focus on the single unit recordings. So we also computed interactions based on joint BSTHs. The long story short is that when we try to understand interactions between the two systems which are not explained by the stimulus locked signal correlated firing rates of the systems, we also during the task found the same interaction patterns, that type 1 dopaminergic neurons are suppressed after cholinergic action potentials, while type 2 dopaminergic neurons tend to exhibit a zero lag symmetric positive correlation with cholinergic firing. During the task events, these interactions were often masked by the signal correlated activities. So, for example, after rewards, the cholinergic type 1 dopaminergic interactions are not following this pattern, which is mostly because of the reward evoked responses of the two systems. But for example, during punishments, both systems exhibited this generally true interaction pattern of the two systems. Next, we hypothesize that if indeed there is a both synergistic and antagonistic relation between the cholinergic and the dopaminergic systems, and it's a characteristic of the two dopaminergic subtypes, then it should be represented in the noise correlations of their response patterns. By noise correlation, I mean the shared response. I mean shared response variances. So more concretely, neurons tend to respond with some action potentials, trial by trial, to cues. For example, here is an example: neuron firing 4 action potentials, another firing 1 action potentials. But in some trials, by chance, they sometimes fire slightly less, or sometimes fire slightly more. In the concrete analysis, these are going to be fixed association tones, so those that are otherwise overtrained on the animals. But still, if the cue has the same value, still there are some variances in responses. And one can fit compute a correlation between these relative responses, that whether when one neuron is firing slightly less, also the other neuron is firing slightly less or not. And we check this for the simultaneously recorded cholinergic and dopaminergic pairs, and find that in all the cases when we find significant noise correlation between cholinergic and dopaminergic pairs, type 1 dopaminergic neurons exhibited always a negative noise correlation, while type 2 dopaminergic neurons were always positively noise correlated with basal forebrain cholinergic neurons, strengthening this idea that even though they are all positively signal correlated, the moment by moment interactions follow this dual nature. So then we try to understand that what can be behind this, what model can be behind this. So we did a massive analysis of cross correlations in the data set, including also the non-identified, non-cholinergic and non-dopaminergic neurons. So these are the cross correlations which I already talked about, the cholinergic and dopaminergic cross correlations. But we also identified other unknown identity neurons too, and we found one group of neurons which seemed to be interesting. So we found a group of neurons which had a asymmetric positive cross-correlation pattern with basal forebrain cholinergic neurons. So they are possibly activated after cholinergic action potentials, that there can be a local connection between these neurons in the HDB. And the very same neurons had a similar suppressive effect on type 1 dopaminergic firing, which was not the case for the type 2 dopaminergic neurons. So we hypothesized that maybe this disynaptic network can explain this segregation of cholinergic dopaminergic interactions. So therefore we made 4 validation, and then we made 4 validation attempts to find proofs that this network can exist and can have a can explain this effect. So first we employed we checked the optogenetic, how other neuron types react during the optogenetic activation of basal forebrain cholinergic neurons. So no surprise, cholinergic neurons are activated during optogenetic activation. We found that these, which I'm going to call putative local basal forebrain GABAergic neurons, are in line with the cross-correlation patterns, are also activated during the artificial activation of cholinergic neurons. And we found a robust suppression of type 1 dopaminergic neurons during the activation of cholinergic neurons. So this.
So how did you label the? Could you go one slide back?
Yes.
So you have you've labeled the GABAergic neurons in basal forebrain?
No, these are. I'm going to call them putative GABAergic neurons because they seem to have an inhibitory effect on dopaminergic neurons. But at this slide, this is just an idea. I will try to prove this on the next slide. We identify this group of neurons based on their behavior during the task, the characteristics of how they react to cues and feedbacks. But this is just guessing right now. But there is a group of neurons which are a good candidate to do this in the HDB. Yeah, but this is not yet proving that it's a strong result that type 1 dopaminergic neurons are suppressed, but this is just suggesting that there might be these local HDB neurons which are the transmitters of this.
And the local GABAergic neurons also express chat?
No, they don't. Yeah.
Okay. Yeah. So those are basically the ones that optotagging doesn't show a response in them.
Yes. So they don't have short latency responses to light. But when the cholinergic neurons are activated for a long time, then they have a slow activation. Yes. So then to prove that whether these whether basal forebrain GABAergic neurons can be the neurons responsible for explaining these connections between cholinergic and dopaminergic neurons, we first made anterograde tracing experiments. So we injected AYFP expressing viruses to the HDB in VGUT CRE mice, and then we examined VTA slices, and we indeed found a massive HDB GABAergic projection to VTA neurons. And then, and by we I mean Dani, my colleague. So Dani also performed in vitro VTA recordings. So he recorded from TH positive dopaminergic neurons and stimulated marked basal forebrain GABAergic fibers in VTA slices, and indeed found that that there were a lot of dopaminergic neurons that were inhibited by the stimulation of GABAergic, HDB GABAergic fibers in the VTA. And then to make this a full picture, we performed the other experiment where we retrogradely labeled HDB neurons that were projecting to the VTA, and then we recorded from HDB slices the activity of these neurons that are projecting to the VTA during the optogenetic stimulation of basal forebrain cholinergic neurons in Chat-Cre HDB slices. And again, we found potential neurons that were projecting to the VTA and were activated by cholinergic neurons locally. Yeah. So this, so indeed, there are GABAergic neurons in the HDB that can transmit cholinergic activity to the VTA. What then we try to understand that whether indeed this connection has a role in learning and in the dopaminergic reward prediction error code. So then we went back to the in vivo experiments, and we performed a chemogenetic suppression of basal forebrain cholinergic neurons. I did this with my colleague Írisz, and very interestingly, what we found first on the behavioral level. So when we suppressed basal forebrain cholinergic activity, what we found, we employed this manipulation after the animal learned the rules. So after the animal were able to perform the fixed associations, and in the session when we first introduced a novel association in animals well trained on the fixed pairings, then we started to employ the basal forebrain cholinergic neuron suppression, and we found that animals were capable to still perform in the fixed associations that the animal already learned. So the animals didn't lose the ability to exploit previously acquired knowledge, but the animals drastically dropped in their capability to learn a new association. So in this figure, you can see that control animals injected with the thread ligand C21 were able to form a new association in practically in one session, while also performing well in the previously learned fixed go tone. But dread treated animals were only capable to perform well on the previously learned association, but were rarely able to learn a brand new association. And this difference also lasted on later stages of the task. So in general, even within animal, so we also had control sessions where we injected saline, and when we compared these control sessions within animals with C21 sessions, we generally found that it took more trials for the animal to form new associations than in sessions without C21 expression injection.
How much do you think this is specific to your paradigm, which is an auditory associative task?
Mm-hmm. Yes. Yeah, that's a good question. So you mean that the auditory functions of the auditory projections of the cholinergic system can have a important role?
Yeah.
Here, why I don't think that that's the main factor, but of course it would be best to test it. But why I would argue that that might not be the most important component is this part here. So the fixed associations are also auditory, and the animals don't seem to struggle much with performing the already formed fixed association. They easily.
Right. Well, I mean, I'm also curious that how much of this cholinergic requirement for forming new associations is specific to the modality of those. Like, do you think if this was a visual task, you would see the same dependency?
I would expect yes. Yeah, and this is not the first result where the cholinergic system was. There are not a lot of results, but I think there are 1 or 2 papers.
And what do you expect? Yeah, go ahead. Sorry.
Yeah, where cholinergic manipulations prevent the animal from forming new associations. Yeah.
And like, how about like in an operant condition, like associations with like pressing a lever?
I would expect yes. Yeah.
Right.
Yeah.
And then you do in your diagram on the left, there are also, it seems like you've been also recording dopaminergic activity at the same time. Are you going to talk about those? Or?
Yes, exactly. Yeah. That's the final question that we wanted to answer, that whether we see that the cholinergic signal is necessary for intact formation of novel knowledge. But what the last question we wanted to answer was that whether it is also required for intact dopaminergic reward prediction error coding. So we had 2 hypotheses. First, that as these signals are signal correlated, and as inhibiting the basal forebrain cholinergic neurons yielded in learning deficits, and therefore rewards were less predicted, we were expecting that this will be reflected in the dopaminergic cue responses as well. And that's exactly what we found, that not only cholinergic cue responses, both to the fixed and the new association were suppressed, but also dopaminergic cue responses were robustly smaller in DREAD-treated animals. And this was also true in the treated animals across Crontor sessions and C21 sessions, that in sessions where the basal forebrain cholinergic neurons were suppressed, dopaminergic responses were smaller to reward predictive cues.
And then this is, so you don't have the type 1. Sorry if I missed.
Yeah, here I don't have it because this is with fiber photometry. Right. So we expect that nucleus accumbens, from where we recorded dopamine here, receives much more type 1 projections, but that should be further tested.
I think I missed something here. So you showed us earlier that the inhibitory neurons in the basal forebrain project to the dopaminergic neurons.
Yeah. Now I have another hypothesis, right, which is here, and that's going to focus on that. So this was, yeah. Let me try to explain this. This prediction of mine was based on the signal correlations between the systems. But as you pointed out, the basal forebrain cholinergic neuron Q responses were the slowest from the three systems or the three neuron populations, which allowed type 1 dopaminergic Q responses within a trial to escape the suppressive effect of cholinergic activation. But this is not the case for feedback evoked responses. So if we check the punishment evoked responses, then the cholinergic activation is much faster than the dopaminergic suppression. So therefore, in the case of punishment responses, what I expect is that when we suppress the basal forebrain cholinergic neurons, then type 1 dopaminergic neurons will be relieved from the suppressive effect of cholinergic neurons on them, and therefore I expect this suppression of dopamine after punishments to be smaller. Yeah, and that's what we found. That both compared between control and dread animals, and both compared between control sessions and treated sessions in the dread animals, we found that the punishment evoked suppression of dopamine release in the ventral striatum was reduced when the basal forebrain punishment responses were suppressed artificially. Did I answer the questions?
Yes.
Yeah. Okay. Yes. So in summary, our working hypothesis is that distributed prediction error codes operate over multiple timescales, and these are not fully distributed by neuromodulatory system. So the type 2 dopaminergic neurons and the cholinergic neurons much more seem to be part of this fast salience-related system, while the type 1 dopaminergic neurons tend to code exactly the reward prediction error, and these systems seem to interact. So acetylcholine and dopamine seem to interact both synergistically and both antagonistically, likely through this synaptic inhibition network. And these interactions seem to be crucial for the intact broadcast of prediction error signals involved in forming new associations. And the last message of our work is that this suggests to us that further future research should focus on considering the two systems rather as a joint network compared to independent networks, including their role in neurodegenerative disorders. So both cholinergic and dopaminergic systems are heavily involved in their malfunctions are heavily present in Alzheimer's and Parkinson's and neurodegenerative disorders with severe cognitive deficits. So therefore, we expect that it should be an exciting future research to examine them as a potential joint network for treatment targets.
Question. So did you do the opposite type of manipulation, that you manipulate dopamine and you see what happens to the cholinergic signaling?
No.
Right, because that would be very important.
Yeah, we don't yet have that.
Right, and then the other question, let's say this radio originates in the cholinergic, that if you had done the opposite manipulation, you wouldn't have seen. Then where do you think the cholinergic is getting this prediction error signals?
Sorry, can you repeat?
So sure. So I'm saying that at this point, it seems like you are saying that the main signaling for prediction errors originates in the cholinergic system, not the dopaminergic system, if I'm reading this correctly.
I would not fully. I don't know. So I believe that both of the systems get largely overlapping inputs, and they might very well both compute, maybe independently, prediction error signals, but from largely shared inputs.
Do you think that these
and
systems
then they interact?
Yeah. Right. I mean, I said that mostly because you did show that if you manipulate cholinergic system, then the signals are diminished in the dopaminergic system. So that's why I said that, and that's why I was curious if you've done the reverse experiment. But then, do you think that these neurons are the ones that are doing the computation, or do you think that they're receiving the prediction error signal?
I pretty much think that, and I think that's yeah, who knows, but yeah, I think that they probably do this. I think they get value related inputs, and then they do the prediction. So I think they do the prediction error computations. Yeah.
We're very much interested in this topic too, because we also study, we study cortical parts, but also the cerebellum. And there also, as you know, there is quite a bit, historically only sensory prediction errors, now more reward prediction errors, and we seem to be seeing signatures of those also in our experiments. So like the link between these neuromodulatory systems and the cerebellum would be pretty curious. Yeah.
It was a good question. So yeah, indeed, the chemogenetic experiments suggest that at least to a significant degree, the cholinergic input is necessary, at least for the type 1 dopaminergic prediction error signal. So why I was confused is that I don't know much about the type 2 dopaminergic one.
Exactly.
Yeah. But for the proper textbook reward prediction error signal, these results indeed very much suggest that the cholinergic input is necessary.
And any ideas about this type 1 and 2 and how so could you link them to other reports of different functional subtypes of dopaminergic neurons in the literature? Anything about the anatomy?
Yeah, probably they can be linked. So type 1 dopaminergic neurons are mostly projecting to the nucleus accumbens. Type 2 dopaminergic neurons largely project to somewhat distinct parts of nucleus accumbens and dorsal striatum. They are also slightly distinct in the VTA, but that's really hard to tell based on our tetrode recordings, because we sample a small we sample population along a line, so it's hard to say very conclusive on this, but there might be a separation in VTA2, but not a clear. So I often have on the same tetrode on the same day both type 1 and type 2 dopaminergic neurons. So it's not a complete anatomical segregation of these systems. The projection pattern might be completely segregated. So yeah, but yeah.
Awesome.
Yeah, but I don't have data on that. Yeah, but based on literature, I would expect that they largely distinct in projection patterns. Yeah.
Okay, that was we're already over time. That was very exciting. Thanks a lot, Balint.
Thank you. Thanks for the opportunity.
Good luck. It's I suppose it's submitted. You guys are in the review or something.
Yeah, we are waiting for review. I'm updating the web page a few times a day.
Well, best of luck. Say hi to
Thank you.
Balázs. Thank you.
Thank
Have
you.
a great
I will.
day. Bye.